Hello everyone, and welcome to your first serving of our new webinar series, The Genome Spot. This is the first time we're doing this style of webinar, and we've decided to leave PowerPoint behind today and open up the floor to some more interesting discussion. I'm very much encouraging participation at home, so if you have any questions or comments or opinions, I'd love to hear them. Either pop them in the comments box below the screen, or I'll address them. Or if you have any questions for the panelists, I'll try to make sure that they're answered. But if you've still got something to say after the webinar, you can also email me at francis.shaw@Frontline Genomics. That's Francis with an E, and I'll forward them on to the right person for you. You can also use the comments box to let me know if you can't hear me or one of the speakers properly, and I'll try and fix that for you. But don't worry if you do miss some of the action, because the webinar is available on demand, as always. So you can watch it again whenever you like, but you can also share it with anybody that you think might be interested as well. We're going to be doing these types of discussion-based webinars on a monthly basis now, but we're going to be covering different topics, so keep an eye out for those on our website and also in your email inbox. If you've got any suggestions of topics or speakers you'd really like to hear from, drop that in an email as well. I'm always interested to hear what's the latest and what's the greatest in the genomics community. So this month's Genome Spot is looking to address a few different topics, but they're all centered around human genetic studies and sample collection. The idea of collecting DNA from your spit is one that we're all familiar with now. It's one of the first things I learned about genetics when I isolated my DNA from my spit with some vodka and some washing up liquid at a school workshop. But it's now something that millions of us are doing at home using commercial DNA testing kits in an attempt to find out who we are and where we come from. But I guess more importantly, what wine we're going to like best. But aside from that, collecting DNA from human samples such as saliva has driven genomic research forward and into places that we never even imagined. And it's also opened up genomics to populations that historically haven't been that involved in genetic research for one reason or another. And our first panelist is someone that has dedicated much of her work to include these underrepresented populations. So Dr. Joan O'Brien is chair of the University of Pennsylvania's Department of Ophthalmology in Philadelphia and is a primary investigator of an amazing study known as the Primary Open-Angle African American Glaucoma Genetic Study. Her achievements have been recognized by a number of awards, including the Woman in Ophthalmology Suzanne Verano Troutman Award, American Academy of Ophthalmology Senior Achievement and Secretariat Award, Laureate Award, and the election to the National Academy of Medicine. Her career is one to be marveled at, but I'll let Joan introduce herself and give you some background of some of the work that she's been doing.
Hi, I'm Joan O'Brien, and the main research work that I do is on a study looking at the genetics of a familial disease, the leading cause of irreversible blindness in the world today, which is glaucoma. Although it's familial and affects African Americans 5 times more frequently and at a much younger age, according to 2009 Nature, only 2% of genetic studies have focused on African Americans. We are located in Philadelphia, which is a predominantly African American city, and we were awarded $11.25 million from the NIH to enroll patients in Philadelphia and study the genetics of this cause of blindness. So to date, we've enrolled about, in the last 4 years of the grant, 9,500 African Americans with full phenotypic data in Philadelphia alone, which is a predominantly African American city. And we've undertaken the discovery phase of a GWAS and now are gearing up for the validation phase of that GWAS, which will then be followed by whole exome sequencing of this population, cases, controls, and people suspicious for disease.
Amazing. Thanks, Joan. So our second panelist is Dr. Alicia Smith, another incredibly impressive woman, but she's looking at a completely different side to genetics. Dr. Smith is Associate Professor and Vice Chair for Research at the Gynecology and Obstetrics. at Emory University School of Medicine, but her research focuses on epigenetics and its role in psychiatric disorders and traits. She's been doing some absolutely pioneering work looking at the development of post-traumatic stress disorder using saliva DNA and following its development in individual patients. It's an amazing study, but I'll let her tell you more about that and a bit more about herself now.
Hi, thanks for having me. A lot of our work is sort of gene discovery using large populations. It's going to be different from Joan's work in that we're not focusing on a specific familial disorder. We're focusing on more common complex disorders, which are more amenable to genome-wide association studies, genome-wide epigenetic studies, and we've been participating in a number of multi-site consortia that are looking to do meta-analyses across multiple cohorts to identify genes that most robustly associate with PTSD and other stress-related disorders. So this involves a lot of multi-omic and comparative research in a reasonably heterogeneous, ethnically heterogeneous group of subjects.
Thanks, Alicia. That's such interesting work. But this type of research isn't able to be carried out without the spit tubes themselves. And our final two speakers today are the kings of sample collection and representatives of DNAgenotek. DNAgenotek are supplying sample collection kits to scientists all over the globe, which is allowing for life-changing studies to be carried out by researchers such as Alicia and Joan. But on the panel today, answering all your questions about how to choose the right kit and the right sample for your study, sharing all that they have learned about optimizing sample collection from working with thousands of researchers and clinicians worldwide, are Mr. Scott Rabuka and Dr. Rafal Iwasiow. So Mr. Scott Rabuka is the director of the Genomics program at DNAgenotek and joined the company in 2008 as the product manager for the Origin product. He's then successfully expanded the product into a complete family of solutions for global genomic research and clinical use. But he's also responsible for the launch of the company's first FDA 510K cleared product in 2011, as well as multiple new products. But I'll let him tell you more about who he is and his role at DNAgenotek.
Thank you, Fran. We're happy to be included on this call, and as Fran explained, I've been with the company for about 10 years in a few different roles. But I think that the biggest interesting aspect for me is just being involved in all the different research projects that we're able to be involved with across, well, I guess across the globe, but also across different disciplines as well. And we've been able to use that knowledge and learning to continue to support our customers as much as possible. And those customers are researchers doing some really exciting work. So for us to be involved with that is a real pleasure. And I guess I would just say we're always happy to help researchers get the work they need to done, and I think we have a lot of experience that we're more than happy to share. So, thank you.
Thanks for that. So our other DNAgenotek representative is Rafal, who is Vice President for Research and Development. He led the team and contributed to the successful 510K clearances of the Origin diagnostic collection device, which is actually the first non-invasive DNA collection device that was cleared by the FDA. But he and his team have developed and delivered new chemistries, products, and services throughout his time with DNAgenotek. But I'm sure he'll be able to tell you more about that and some of his other work too.
Thanks, Fran. Yeah, as Fran said, I lead the R&D team here, and beyond just designing new non-invasive, intuitive devices and the chemistries that go along with it that some of you may be familiar with, and we'll be discussing some of the studies that use them. One of our focuses is also to continually stay current with the technology evolution, So we heard already, a comment that, studies will be used on arrays and then exome sequencing, and that's something that we continually do, and we have a lot of information that we can share on usability of the kids on a very large spectrum of technologies.
Amazing. Such interesting work. But yeah, before we start the discussion, something that we're trying today is audience involvement. So you people listening at home, I set you a couple of questions that you can answer at your desks. But I'll give you a heads up when those questions are going to appear on the screen. You can answer these if you want to, or you can just sit back and listen to our speakers today. It's entirely up to you, but we do encourage it. So firstly, I'd like to ask a question to Joan about some of the key challenges that you face when you're both designing the studies and carrying out studies, such as the primary open-angle African American glaucoma genetic study. Hello, Joan?
Oh, I'm sorry. Do you hear me now? Sorry.
Yeah, sorry. I think you cut out there a little bit. Did you hear the question?
I did. Thank you. So the main challenge has been, first, recruitment of a large number of African American cases, controls in a single city to obtain sufficient power for gene discovery. And it's been important to reach out to the community, and we've had the good fortune of having the community reach back to us. So leaders of African American talk radio, such as Word Radio in Philadelphia, have been very involved in interviewing participants in the study, as well as our African American and glaucoma specialists who recruit for the study. We've put advertisements in subways and bus stations. We've had outreach events. We've obtained money from other studies to do full phenotyping on individuals, because we believe that this disease is probably many smaller diseases, and we need the full phenotypic information. And we've been able to recontact patients to bring them back in when we find an interesting variant. So I guess those would be the main challenges in this recruitment.
Yeah, definitely. When we spoke about it previously, it was amazing to hear some of the other motivators for getting that recruitment that you tried out and were really successful in doing. Alicia, what are some of the things that you keep in mind when you're studying the epigenome and psychiatric traits? Do you face similar challenges, or are they challenges that are specific to your studies?
We do. In addition to working with people that we recruit locally, one of the things that we like about using saliva DNA is that we can collect in the field. So just yesterday, I was having a conversation with a collaborator from Peru who is wanting to do a longitudinal study of a birth cohort that's been ascertained in Peru. But one of the issues that we have is going out to more remote villages. It's difficult to have access to phlebotomy tools. It's difficult to have access to ways that one can longitudinally sample participants that don't require specialized training and that don't require specialized storage. And we like saliva from that because, one, people agree to spit in a tube a whole lot more readily than they agree to be poked by a needle. But also because we can get a more comprehensive group of people from places where we wouldn't if we weren't collecting saliva.
Yeah, definitely. I think people are far more comfortable now, especially with some of the latest sort of commercial kits as well. It's something that we all hear about now and that we're really familiar with. But something that DNAgenotek do, they offer many different kits, not just saliva kits, that allow for collection of samples and all different samples. What are the most important things researchers and clinicians need to consider when choosing a particular kit or biological sample to use in their studies?
One of the things that we think about for epigenetic studies is cell composition. Because methylation and epigenetic marks vary from tissue to tissue, it's important to sample from a tissue that is closest to or representative of the tissue where you are looking for an effect, or something that's consistent biologically with your hypothesis. And so that's a lot of the issues that we have with respect to picking a tissue. If we have an immune hypothesis, we're interested in blood. If we have a hypothesis related to PTSD or the brain, or that requires longitudinal sampling, often we pick saliva for that, just because of its properties and similarity.
Yeah, it's definitely something to keep in mind. We need to keep studies as similar as we can as when we're trying to study those samples that are really difficult to access. So I guess this one would be for the guys at DNAgenotek. Is this some of the things that you consider when you're offering advice to researchers and clinicians, or is it you take a different approach?
Yeah, if I can start a little sooner, I think one of the issues we commonly will see is that researcher will plan their study and will think about what do they want to answer in the end, rather than how are they going to get to that answer. So quite often we engage with researchers, and they haven't thought about how they're going to even get the sample. So there's so many aspects of sample acquisition that can be challenging, but as was just commented, you really need to think about what question or questions you want to answer and how you're going to get there. So that that comes into play with sample types and analytes, and then also sufficient sample to be able to do what you want downstream. And you have immediate goals for your research study, but you need to think about what you want to do longer term as well. So we talk about future proofing your research by collecting a sample that will be sufficient for your immediate needs, but also used for future needs as well. So, Rafal, I don't know if you want to add some comments to that.
No, I think I just added. I think it's been touched on, but it's important to maintain consistency in your studies, right? And as Scott alluded, it's kind of future proofing. So if you've collected a sample that you can store and use for future studies as technologies evolve, there's a benefit there for sure. And as far as epidemiology, I mean, we really rely on experts, like Joan or Alicia, to demonstrate these use cases. We do internal work, but we rely on them to publish and share that knowledge. And I think we are seeing from them and from others that when you're looking at epidemiology, especially cases of brain conditions, saliva is a great source for that.
Yeah, it's really interesting how we can now use saliva DNA to study the brain. It was quite a remarkable research to find that it's so similar. So my next question is for listeners too. So if you're listening at home, you should see a question pop up on your screen. If you want to, you can answer this question. If you don't, that's absolutely fine as well. But we'll do this in turn. So, Alicia, what sample collection techniques are you currently using in your studies? Is it only saliva samples, or are you making use of other samples as well?
We have a number of active studies. A lot of them use blood. Some of them use tissue samples. We have a study of pediatric Crohn's where we're looking at rectal tissue. We're also using a lot of saliva and some buccal swabs. And the studies that use buccal swabs over saliva is because we're looking for to use that DNA both for microbiome studies to make sure we have adequate bacterial DNA, but also for human studies. And in the studies where we tend to use saliva, we've noted we use Origin kits. Shout out to Origin, love your product. But also we use it because we get a much lower proportion of bacterial contamination when we use that in much higher amounts of DNA. And so for epigenetic studies with saliva, we can use bioinformatic methods to estimate the proportion of buccal epithelial cells relative to leukocytes and use that as a covariate. So we can sort out a lot of the technical heterogeneity, and we've found some really exciting results with that. Yeah, sorry, just responding quickly to a question that I saw on the question feed. People were asking about the difference between buccal and saliva, and we do use buccal. In general, we find that there is a lower yield for buccal swabs relative to saliva. And so just from pragmatic reasons of wanting as much DNA as we can possibly get for our studies, we tend to prefer saliva. But buccal is certainly reasonable to use as well.
One of the areas of feedback that we had early on was for patient populations where spitting just wasn't possible. So we have some sponge-based solutions that will essentially absorb the saliva and whatever cells they can get at in the mouth, and then the stabilizing chemistry would provide the same properties as Origin would. So back to the question around when you think about sample collection, you also need to think about the population you're collecting from, not just location, but also what they're capable of providing. So we have some elderly cohort studies that spitting is just very difficult, and also pediatric studies where you'll have a 1, 2, or 3-year-old that again spitting is difficult.
Yeah, that's really interesting, and it's great that you're sort of opening up genetic research to both the elderly and children as well. That is going to be less likely that they're going to give other types of samples. We're actually getting quite a few questions in on the question and answer. One of which is for Joan about her studies. So I'll ask that now. So Joan, there's somebody here that's also collecting saliva samples from African Americans in a longevity and stress study. And they're wondering if you're planning on performing methylation chips as well as GWAS.
At the present time, we're not funded to do that. It's very interesting, and I would love to do that in a follow-up study. But at the present time, we're funded to recruit 10,000 African Americans in this city, and then complete our GWAS, and then do whole exome. And so we really don't have the resources to do methylation chips at this time, but I think it's a very important question, and I would love to be able to do that.
Great. So one of the other questions we've also got, which is really interesting and not something that I've thought of, but is there financial incentives for samples? If so, what are the concerns of this incentive affecting the results? And if this does happen, how do you handle it? I guess this would be to either Joan or Alicia, whoever would like to jump in first.
Well, I think with any scientific study, there's a concern that remunerating people for their participation could be coercive, and that's something that IRBs look specifically at. We always tend to pay a small amount for participation in our study. usually on the order of $20 or so for something non-invasive, which I don't think anyone would consider particularly coercive. Joan, what do you do for your studies? Joan, if you're speaking, you're on mute.
I'm so sorry. I'm so sorry. I am speaking. So we give a $10 gift card, and our IRB has not felt that to be coercive, and I don't think that it is a sufficient amount to affect the outcome of the study.
Okay. Yeah. So I do remember you mentioning some of the incentives. What other incentives did you also give? I know that you had some sort of kind of like a celebrity involvement, Joan. How successful was that, and could you maybe explain that?
One of the voices on Word Radio is a very well-known African-American newscaster, and one of our challenges in the study was that we were having, as many studies do, more women enrolling than men. When we had the advertisement, which is every hour on the radio, and it was a very familiar male voice, although he didn't identify himself, he was recognizable to this community, and then we saw a large uptick in calls to enroll in the study from African American men.
Yeah, it's a good incentive, and to get people involved in different ways is kind of creative in a way. So one of the things that the people are concerned about are, and there's been some debate over whether saliva DNA matches up to DNA from other samples. How important is the quality and integrity of the DNA in each of your studies, and is saliva matching up, I guess? Alicia, you could maybe answer this one first.
In general, we find that relative to something like blood or fresh tissue, that there is more degradation in saliva DNA. That comes in part because when you're using blood, those are all, for the most part, live cells, versus saliva DNA includes a lot of buccal epithelial cells, and the cells towards the outermost layer are those that are more likely to be dead. And because of that, we see a little more degradation, but not so much so that it's not of sufficient quality to use for microarray analysis, targeted genotyping, and even next-generation sequencing. So we have found...
Is it similar with you, Joan?
Yes, so we actually published a study with Illumina before we switched for the first 2,000 or so samples, we used blood. And then, to reach our goal, we wanted to discover if we could get by with saliva. So there are still studies that we're doing with blood, such as functional studies of genome-wide significant variants. But in terms of encouraging recruitment, it was much easier for us to prove with Illumina scientists that our call rate was essentially the same, and it was high, 99.6% call rate using blood versus saliva. And we did publish that with them, and that allowed us to switch for most of our recruitment to saliva. The amount of RNA-free DNA is sufficient for us to do array-based genotyping and also next-generation sequencing in our lab, as well as whole exome sequencing. So it is sufficient, and it provides us with less DNA as well, and perhaps slightly lower quality, but for the purposes that we need it, the saliva DNA is fine. Once we need larger amounts of DNA for, say, trying to evaluate a variant of significance and understanding what that actually does, then we do start to recall patients and use blood as our DNA substrate.
Fran, if I may jump in.
Yes, of course, go for it.
Yeah, I just want to add, I completely agree with Joan and Alicia's views. Just want to add a little bit of a technical view. Agree that buccal cells are the ones that are lower quality. They're the ones who are shedding off. That's why, DNAgenotek's focus has always been on saliva. Even our swab-based kits are looking at absorbing saliva. There are some nice publications out there looking at bone marrow donor recipient studies that show that up to 70% of cells in saliva actually are secreted actively from the blood, and those are quickly, in minutes, recirculated. So we're focusing on that large population of nice, healthy cells for collection.
Okay, yeah, great. Well, I guess it kind of answers my next question, but I was going to ask how DNAgenotek were actually ensuring that the quality of the sample is maintained, I guess, throughout transit and throughout the research as well.
That's definitely a large focus of what we do as we design our devices and chemistries. A lot of effort is put on both the physical components and the chemistry components. They go through very rigorous testing throughout, and we maintain that testing actually after launch. So, on a yearly basis, we monitor that our devices continue to meet those specifications. And back to, related to the quality, we have shown also internally, and it is on our website, many different white papers comparing blood to saliva, from arrays to exosomes to whole genome sequencing. And, as we heard earlier, we see the same thing internally, concordance rates well above 99%.
Okay, great. Well, the next question is actually for you guys at home as well. So if you're wanting to answer this question, please go ahead. It's more of an opinion question, but I will put it out to our speakers as well. So in your opinion, what needs to be or can be changed to improve your results, or to make them more reliable or more informative in terms of sample collection or just the way you carry out your studies? I guess, Joan, you could maybe answer this one first.
I think what's been important for us, because our patients are elderly, is to make sure that we really work with them to make sure we have sufficient sample size of saliva in order to make sure that we have sufficient DNA. We will sometimes use a little bit of sugar or a little bit of lemon juice to help with the saliva collection, and that was a suggestion that we actually got from DNAgenotek. And that has, we do it fairly rarely, but when we need to, we do. But you definitely need to make sure that the patients are able to provide the whole amount of saliva that the kit requires.
And how about you, Alicia?
We haven't noticed that much of a problem, and it may be that we work with a younger patient population, but we have not noticed anyone not be able to provide sufficient sample. And sometimes volumes are a little bit low, but we've noted that yields are still fine. I can say from personal experience, every now and again when we need to train somebody in extraction procedures, or when I did my 23andMe results, I've provided a sample in just a standard Oragene kit, and it's a lot of spitting. It takes a good 5 minutes versus a very strange sort of experience to get a DNA sort of enough saliva to fill the kit. But a lot of people still just prefer it, and other than it being a longer time commitment, that we haven't noticed any problems.
Okay, and how about you guys at DNAgenotek? Is there anything that you're working on to sort of improve, or you've noticed that you're getting some feedback and something that you're working on, or that is the new kits or available?
Yeah, well, one of the areas, and maybe I'll talk about one of the research studies we work fairly closely with, and that's from the Simons Foundation, their autism cohort study. So they're aiming to collect 50,000 trios for autism research, and as you can imagine, can be a difficult population to collect from. So we've spent a lot of time working on customized instructions and helping with collection videos and so on, and even different collection formats to make sure that when they get the chance to collect the sample from that patient and individual, that they get a sample that's going to be usable for their study and downstream. So that's kind of a specific case, but I think the point is, when you're looking at different patient populations, you need to think about their environment and the context in which they're collecting in. If they're going to be collecting at home by themselves, or if they'll be in a clinic, or if it's supervised, all those aspects come into play when you want to make sure that they have the support they need, whether that's video or pictorial instructions to ensure that they can collect a great sample. And if it's a clinic, if it's a clinic collection, then you want to make sure that the nursing staff or the clinicians that are collecting those samples will be trained to have the tools to be able to help the individual collect. And then another aspect of our of collections is again, with the context of the patients, English may not be their primary language, so you want to make sure that they have instructions in their native tongue. So a lot of those types of things, some of them might seem minor, but they all enrich the collection environment and ultimately lead to successful collection.
Okay, thank you. Sorry.
This is Joan. We use clinical trial coordinators who are from the pre-med programs at Bryn Mawr and UPenn. Most of them are students who weren't pre-meds, they're post-baccalaureate students who have a year or two to take courses and take their MCATs. And so they're very much wanting to be with the doctors and with the patients. And they stay with the patient throughout the saliva sample collection and encourage them. And many of the clinical trial coordinators, besides being pre-med students, are also African Americans from our community who can use the Penn tuition benefit to pay for their classes. And so there's a very good bond that develops between the clinical trial coordinators and the patients, and the clinical trial coordinators are very much involved in talking them through the process of collection.
Joan, if I could add to that, your comment made me remember one other issue that we have had in which results could be improved on. We did a study remotely where we were prospectively characterizing people, and we gave them a group of kits to take home and do self-collection at home. And so there wasn't a clinical coordinator watching and supervising collection. And we found a number of people said that they gave a sample and there was just no DNA in there. And so some sort of quality indicator or qualitative indicator of whether or not there was definitely a sample provided might help with at-home or remote studies in which you can't get coordinator time.
No, that's a great point.
I think we've got a question here for you, Joan, about the saliva production aids that you're using, i.e. the sugar or the lemon juice that you said. They're wanting to know whether it actually increases the cell yield as opposed to just diluting the sample.
I haven't looked at whether it increases the cell yield. It does increase the saliva yield in the rare patients who can't do it on their own. We don't do it a lot, but if we can't get a patient to provide saliva to the full amount of the kit, then we will use that technique. But I haven't done an analysis of how much is saliva and how much is cells.
I can just add to that. The studies we've done here at DNAgenotek actually would suggest that any kind of stimulation, whether it's sugar or lemon juice, has the potential to actually slightly decrease the yields per volume. However, overall, your collection is much more successful because you have collected sample. What happens biologically is you stimulate serous saliva, so it's like more watery, and the amount of cells coming through is actually lower per volume. But again, you can actually collect the sample, a large volume of it, which overall gives you a successful collection.
We've had very few instances at all where we haven't had sufficient sample. But I do think the presence of the clinical trial coordinators throughout the process is key to that, and the encouragement they provide.
Great. So another question that we've had in from one of our viewers is about whether or not saliva-based testing is ever going to get beyond the point of just being good enough. Is it actually going to be able to replace invasive procedures or plasma for DNA collection? I guess I'll open this to the floor if anybody wants to jump in.
Well, it's been good enough for us in terms of being able to do, as we mentioned before, array-based studies, next-gen studies, whole exome studies. It's been sufficient for those. The problem, if you can't recontact patients, and we've been able to, is that the amount of DNA that you obtain would be less than if you obtained a blood sample. And Alicia has already talked about the relative benefits of different kinds of samples for different kinds of studies. But what we found is that the patients in Philadelphia who've agreed to be part of the study also are quite willing to come back if we need to do blood collection for, say, a functional study of a gene to make sure that we understand that if a variant is significant, what in fact does it do? Because the point of any study is eventually to help the patients. And so the goal of this long term, and we're about to put in our next 5-year renewal, is to understand what variants identify with what phenotypic data. And we have very detailed phenotypic data, and that will allow us to not think of this leading cause of blindness as one giant disease. We'll clump and we'll put together that for power, but we'll also subdivide and find out if there's different variants that are associated with different phenotypes. We've already been able to do that in a masked fashion. And it appears that this is a disease sometimes of degeneration of the optic nerve as a primary optic neuropathy, and other times that high pressure in the eye causes the nerve to degenerate. And those are probably very different kinds of genetic variants. So I guess there are reasons why you would want to use blood for more advanced studies because there's just more volume of DNA there. But it hasn't been a problem for us to use saliva for all the purposes that Alicia and I have discussed. And when we need blood, we just recontact.
I guess it's also important to point out cases where saliva is already the frontline diagnostic. issue, and not just a good enough. The direct-to-consumer genetic testing like 23andMe and Ancestry.com all use saliva DNA as their first choice.
So I think, Alicia, do you agree with me? Depending on the study, for our purposes, for getting high-quality DNA, it's not just good enough. It's actually comparable in array-based studies, at least we've proven, to blood.
Yes, very comparable in terms of quality. In terms of, specifically for genetic studies, but also for epigenetics, it gives an additional layer of dimension and is first choice for a number of people from that as well. There's also a number of, there's a really active area of saliva diagnostics where people are looking at saliva diagnostics, both protein and DNA and methylation, with respect to head and neck cancers and certain things that are really relevant in the mouth as well.
No, that's an excellent point.
Yeah, we've developed a product that's on the market for stabilizing RNA from saliva, and when you think about that, well, what are the applications? And really, it's been very specific to applications like mental health and head and neck cancers and so on. But it's again, it's specific to the source.
I think that's a very interesting point, and I think it may have broader applications with time. I think as that gets better known, looking at the transcriptome may be very important.
Thanks for that, guys. I don't know if you guys at DNAgenotek have seen the question on the question and answer board, but it's asking about the stability of the saliva samples and whether or not you're looking at using solid stabilizers. I don't know if you'd like to address that question.
Sure. So our kit is designed with a liquid stabilizer. We believe it provides benefit for a usability standpoint, especially once it gets in the lab. Many saliva samples are very viscous. Our stabilizer and chemistry ensures that the viscosity is gone, so in cases where you actually have high throughput, you can use them in an automation system without any concerns. Just facilitates the use case. We certainly could and have looked at dry options, but we have found those to be inferior in our case.
We haven't found saliva samples over the last 4 years collected in the manner just described to be unstable. We can continue to extract DNA of high quality.
We actually, if anyone's interested, on our website, we have a number of posters that we've presented. We make claims on our research use products 5 years post-collection stability, and we actually have posters where we've gone way beyond that, showing that you get excellent results from arrays and sequencing, and the concordance between those two technologies is well in excess of 99% when using the samples.
Alicia, we've also got a question that I think maybe you might be able to answer. It's asking about whether or not anybody has any experience using a pediatric buccal kit. And I know that you have or have been involved in some studies with children. Is that something that you're familiar with? Have you had any issues with that if you have used them?
Yes, the pediatric kit is the one that was alluded to earlier in the call, where there's more of a sponge. As babies don't spit particularly well, you largely sponge up the saliva. and then spin it down and extract from that. And we've found it definitely has lower yield than, let's say, a full-size adult kit, and that's proportional to the amount of actual saliva that you get from it. But we still find the yields are higher than buccal swabs taken from the same children. So it's, I'm really happy when our kids learn to spit. Their parents may not be, but I am. And but the pediatric kits are fine. And I don't know if well, you know, it's the pediatric kits are good enough. Is basically where it is. It's going to be better than any of the alternatives, and even better in terms of yield than a heel stick and a blood spot, which is, I think, what a lot of people also do with children.
That's funny that you say about the spitting. That's funny. But yeah, Joan, we've also had a question in for you as well, to do with the how you said that saliva samples were stable after 4 years. They're wanting to know how the samples were stored.
I refrigerate them, I believe. I'm not the best person to tell you that because I'm technically not in my lab as much now that I'm a chairman, but we haven't had any trouble, and I would bow to Alicia about what she does, because I can't absolutely guarantee that that's what my lab is doing.
Typically what we do is we store at room temperature for until we get an appropriate batch size, and the folks from Genotek could probably say this more precisely, but we find that the saliva kits are stable at room temperature for up to 6 months or so. So we batch them and send them over to the lab where we aliquot them into smaller, 2 ml tubes that we use for extraction, and then we store them for long term at -80.
Yeah, I could add to that. Our 5-year claim on our research use only kits is unstabilized in uncontrolled room temperatures environments. And I alluded to some studies where we've gone way beyond that, many years beyond that, and those were, again, uncontrolled room temperature conditions. We actually recommend room temperature over fridge for 2 reasons. One is space, so fridge space is expensive and not necessary. And second, it's actually easier to use those samples. Nothing precipitates out if using at room temperature.
Okay, great. I think we might have time to squeeze one more question in, but we are rapidly running out of time. But one of the questions that, again, has come from viewers is asking if any of you can talk about blood spots in terms of yield and quality of DNA as an alternative to saliva.
I guess I'll answer that one because I was the one who brought up blood spots. Typically for our studies of newborns or young children, blood spots from a heel stick or a finger stick onto Whatman paper or FTA paper is typically what's done. That too is stable long term at uncontrolled room temperature and usually have reasonable DNA yield. I think the difference in it is the amount of cells that you have to work with. Average, each blood spot is something like 100 or 200 microliters of blood, which only has so many white blood cells in it for the purpose of extracting, whereas something like saliva has more cells proportionately, and you tend to get higher yields if you're using one of the full-size adult kits.
Okay, great. Thank you for that. But I think that's actually all we have time for today if we're going to finish on time. But I'd like to just say thank you so much to Alicia and Joan today. I don't know about you people listening at home, but I've loved listening to their experiences and about some of the studies. And I've also like to say that it means a lot to me, as Joan and Alicia have both helped me shape this discussion and the webinar right from the beginning of my own research to help put this new format and Frontline Genomics first ever webinar as part of the Genome Spot series. So I really appreciate that. You've both been really helpful, and I can't thank you enough.
Well,
but I'd also
happy.
like, oh, thank you.
We're very happy to do it.
Well, yeah, I'd like to also like to thank DNAgenotek themselves. It wouldn't be possible without them. And for having their speakers here today and representatives to give all of their experience and knowledge about all of the different types of sample collection techniques. But if you'd like to know anything about their products or recruitment or have any further questions for them, their emails are on the screen now, so you can drop them an email. But something else DNAgenotek are also doing, they are kindly offering a complimentary sample collection kit to all of our listeners today. So all you need to do is email the information box with webinar sample kit in the subject line, and they'll sort that out for you. So that's really generous of them. But yeah, thank you again, Scott and Rafal. It's been a pleasure to have you today.
Yeah, thank you. We're happy to be a part of this and really enjoy the discussion. And I would just say we love talking to researchers and their customers, so reach out anytime. Thank you.
Yeah, no problem. So for you people listening at home, if you did find this interesting or helpful, please let me know. I also like to hear from you. So drop me an email giving me your opinion or any comments you have. As I said, this is the first time we've actually done a panel discussion webinar, so I'd love to hear your feedback. There's actually a survey after the webinar if you'd be kind enough to sort of stick around and answer some of the questions there. And if there's anything that you missed or you want to go back on, remember that it's also available on demand, but we'll send you a link in the email for that. But yeah, thank you so much for listening, and I hope you all enjoy the rest of your day. Okay, I think